So thank you. It's my pleasure to be here today and to see everybody's face on the screen. And I'm honored to be able to speak to such a great group because I read some of your bios online and quite impressed by the work that that your group is doing. I'd also like to do my own land acknowledgement. I'm originally from Newfoundland, but I make my home and work in Calgary. The City of Calgary and the University of Calgary are located on the traditional territories of the people of the Treaty 7 region in Southern Alberta. We're also home to the Métis Nation of Alberta Region 3. So these are my disclosures. I have received funding to the Canadian Hepatitis B Network from industry sponsors. And my group, my colleagues and I in the liver unit do a number of clinical trials on liver disease, and I'm either a PI or a co-investigator. We just finished the International HBV meeting in Toronto, and I'm using resources from that meeting, so it's very timely. And I'm also using resources from the European Liver Meeting and the American Liver Meeting, and I'm happy to share my slides. So let's just jump into what I'd like to cover today. So I'd like to start with why we need a cure for hepatitis B, despite having very potent or highly effective antiviral therapy. I would like to provide a brief overview of molecular biology and immunology of hepatitis B, and then jump into reviewing the new drugs that have advanced to clinical trials. So I'm not going to talk that much about the preclinical drug development. And there are two main classes, which are directly acting antivirals and immunomodulatory therapies. And then finally discuss what the so-called experts are saying about what we need to have for a cure in terms of combination treatments. I have lots of time for my talk today, apparently, so please jump in and speak up if you want to interrupt and ask any questions. So I'd like to keep this pretty informal, even though it's sometimes difficult with these Zoom type of talks. So why do we need a cure? So this was a slide from the president of the Hepatitis B Foundation at the HBV meeting. Despite the fact we've had a vaccine for 4 decades, there are still 300 million people living with hepatitis B. There are still over 6 million under the age of 5. So this is despite the recommendations for providing birth dose or infant vaccination. In 2019, there were over 1 million new cases and 820,000 that died from hepatitis B related causes. The vaccine has been available for a long time, but it's important that we give it at birth dose or in early infancy to prevent transmission, because the risk of becoming a chronic carrier or living with the disease lifelong is greatest in those under the age of 1. And I'm putting a plug in for that because this is sort of a key goal of the Canadian Hepatitis B Network. In 2020, the goal, according to the WHO, was to have 80% of people diagnosed, but there's only about 10% of people that know about their disease. And even less of the total, only 2% have been treated for hepatitis B, which is important because that will reduce your risk of liver disease and liver cancer. So we have a long way to go before we meet the WHO elimination goals. And as I mentioned, even though this talk is about cure, I'm going to put a plug in for prevention. The Hep B network and colleagues within the liver societies, I should have asked your group as well to sign on our letter. We sent a letter to the Minister of Health, the chair of the National Advisory Committee, and the Public Health Agency of Canada to advocate for universal birth dose vaccination in Canada. You might be surprised to know that 5 provinces, including Ontario, which has 14 million people, do not give the vaccine until pre-adolescence or grade 5 or grade 6. And this is shocking because there was a study done by Jordan Feld last year that showed that there were dozens of children, I think 140, born in Ontario, diagnosed with hepatitis B when before they even were eligible to get the vaccine. And so we need to push our policymakers to implement the vaccine. Unfortunately, the letter that came back was very polite, but very non-committal. And they said, it's up to the provinces to decide, blah, blah, blah. I think we need to send our message to the National Advisory Committee on Immunization to advocate for this. I think this is an important thing we should do. You know, the WHO keeps track of who does birth dose vaccination, And Canada doesn't even show up on their tracking because we don't do it. So it's actually quite shameful and embarrassing for our country not to do this. So that's it for my plug for prevention of Hep B. So why do we need a cure? So the scientific community has agreed on certain definitions. They define a partial cure as people that have a normal liver enzymes, low-level viral DNA, but they still remain surface antigen positive on a nucleoside analog or antiviral therapy. This is still good because it will reduce your risk of liver disease and end-stage liver disease. A functional cure is the current goal. In this, a patient has achieved loss of surface antigen and developed antibodies. Unfortunately, it can take more than 40 years on a nucleoside analog. Some people, especially adults, if they become infected as an adult, will lose surface antigen and develop a functional cure. But the virus can still persist within the cell nucleus, and this is the reason why they would be at risk of reactivation with immune suppressing therapy. And I'll talk about that a little bit later when we discuss the molecular biology. A sterilizing cure is where there's complete viral eradication, and this is not currently achieved by our current treatments. Hepatitis B is a dynamic disease. It has a complex life cycle. It replicates via an RNA intermediate similar to HIV and hepatitis C, and thus it has multiple genotypes. So combination treatment will likely be needed to achieve a cure. We did a study with the Hep B network recently looking at the benefits of a functional cure, because there's some argument to say, well, you have a nuke, you're on long-term treatment, that's good enough. Do we really need to advance the envelope for treatments? And in our study, we looked at people that had achieved surface antigen loss versus those that had persistent surface antigen. And we found, acknowledging it's a retrospective study with the limitations, that the risk of end-stage liver disease, liver cancer, and cirrhosis were lower in people that had achieved surface antigen loss or a functional cure. And this is experience from my clinic. I'm sure many of the clinicians who treat hepatitis B have had similar experiences in that people that have been on long-term therapy for decades with suppressed viral load, very low level antigen, can still relapse and have very severe hepatitis flares and viral load flares after stopping treatment. And I highlight this person in red because her viral load was undetected. Her surface antigen levels were almost negative. We stopped treatment, and within a month, her ALT had flared to dramatic levels with a viral load flare, requiring restarting her antiviral therapy. Fortunately, all of our patients have responded to treatment, but it really highlights the need we need to get better therapies for Hep B. So I'm going to go through this life cycle for hepatitis B to mainly highlight the key steps that are being targeted for the directly acting antiviral therapies. And I've taken this from a nice review that was written by my PhD student, and she created this nice figure for her thesis. So the Hep B binds to its receptor. It's actually a bile acid transporter. For decades, nobody had a clue how the virus was able to enter the cell, and it was only discovered in 2012. So it uses the surface protein to bind to its receptor. It then delivers its genome into the nucleus of the cell, so-called covalently closed circular DNA, which is the master template. It hides within the liver cell, and it is the template for all of the RNA transcripts. Following that reverse transcription of its RNA, it then forms its capsid or core. It assembles its envelope within the endoplasmic reticulum, and then leaves the cell to go on and infect new hepatocytes. Hep B does two additional things which are interesting in its life cycle, in that it also integrates its genome into the host DNA. And this is the reason when you hear of Hep B, you need to think of liver cancer, because this is one of the mechanisms why Hep B patients are at increased risk of liver cancer. The other interesting thing it does is that it produces non-infectious subviral particles in excess, especially the surface antigen. And it was a bit puzzling as to why it would do this in its life cycle, but it's believed that this would serve as an immune decoy in and also contributing to immune exhaustion or inability to achieve clearance of the virus. As I'd already mentioned, Hep B also uses reverse transcription in its life cycle, and that is why you can have presence of variants or genotypes because of the error-prone mode of replication. So when we talk about the different drug targets that are in development, these include entry inhibitors that are targeting the binding to the NTCP or bile acid transporter, those that are targeting the RNA inhibitors or antisense oligonucleotides, those that are capsid or core inhibitors preventing encapsidation or causing premature melting or disassembly of the capsid. We know about the nukes that target reverse transcription, and also those that prevent secretion of the subviral proteins. This is a busy slide, a bit complicated. I'll let you take a moment to digest this, and if you have any questions, please jump in. Okay, all good. So I mentioned about the cccDNA. So this is the menace. This is the reason why the Hep B can persist for decades, even after you lose surface antigen, why we have to keep it in mind when we're giving patients immunosuppressive therapy or cancer chemotherapy. It is the first viral product. It's actually constantly replenished. So it keeps reforming new molecules through a recycling method. It was believed that the half-life was quite long, about 6 months, but there was recent intriguing data just presented at the meeting in Toronto showing that it could be much lower with cell division. It's not affected by the nukes. The nukes target the viral polymerase or reverse transcriptase inhibitors. The other disadvantage is that the interferon that we have used for decades also for hepatitis B doesn't directly affect cccDNA. It has more of a non-specific limited antiviral effect. So the new treatments need to block both replenishment as well as new infectious virions from being produced. So when I talk about hepatitis B immunopathogenesis, it's, as we should recognize, it's also an immune-mediated disease. And in order to achieve clearance of hepatitis B, you need to have an efficient, innate, and coordinated adaptive immune response with production, with activation of CT8 T cells and B cell help that producing antiviral cytokines to kill off infected hepatocytes that contain cccDNA, as well as producing and neutralizing antibodies to prevent infections of new virions or new hepatocytes. And thus there is quite a lot of activity in development of new immunomodulatory therapy in order to achieve hepatitis B surface antigen loss or a virological cure. I'm just keeping a note in the chat box. Okay, yeah, there's nobody talking to me there. Good. Sorry about that. I'm trying. Okay. So I'm going to go through the directly acting antiviral agents that have progressed to sort of late or mid-stage clinical development. And this is a nice review published in Gastroenterology that reviews all of the different agents and their different stages of development. I'd like to point you to this website. It's run by the nonprofit Hepatitis B Foundation, and they keep this more up to date if you look under Drug Watch. It also contains a lot of resources for your patients. So if you're not aware of this, I would encourage you to show it to your patients so that they can see what's available for them. So these agents also include the entry inhibitors, which was recently purchased by Gilead. It's called bulevirtide. I'm going to call it Hepcludex because I can't pronounce it very well. Capsid inhibitors, the antisense oligonucleotides are RNA inhibitors, surface antigen inhibitors, or those that prevent surface antigen secretion. I'm not going to talk about the preclinical agents in terms of gene editing or those trying to destroy or repress HPV DNA or cccDNA, but this is really the holy grail, right? This is what we need to achieve a sterilizing or virological cure. So I'm going to talk about Hepcludex first because I think this is the one that's going to be approved within the next 18 months, hopefully, for hepatitis B and hepatitis delta virus co-infection. I don't go through hepatitis delta in my talk, but as you're probably aware, this is a so-called satellite or parasite virus, in that it uses the hepatitis B surface to complete its life cycle and thus enters the liver cell by the same mechanism. And people that have Hep B and Hep Delta co-infected are at very high risk of liver disease, cirrhosis. It's the most severe form of viral hepatitis in humans. So this viral entry inhibitor, which targets the bile acid transporter and the NTCP, has been used for Hep B and Hep Delta. The dose is anywhere from 2 to 10 milligram daily. Because it targets the bile acid transporter, it can cause elevation of bile acids, but it's only at a lower concentration, and so it hasn't actually affected the transport function of the transporter. It has been conditionally approved already by the European Medical Agency for hepatitis delta, because delta is an orphan drug with very limited treatment options. And I believe it's going to be submitted soon to the FDA for approval. So I'll talk about the two main clinical trials, the phase 3 and that has been published, that has led to the approval by the EMA. So this was recently presented at EASL, and it looked at the bulevirtide monotherapy at low and high dose in patients with hepatitis delta. Their primary endpoint was undetectable hepatitis delta RNA, as well as liver enzyme normalization. And what they found from this phase 3 study, that 24 weeks of monotherapy was associated with significant decline in delta RNA and improvement in liver disease or ALT activity. It was very well tolerated in people that had compensated delta, and these findings are further support the conditional approval in the European Union. It has not been used for hepatitis B monoinfection yet. It's just under preclinical development. But given the fact that Gilead has recently purchased the drug, I'm expecting some studies to come through. But this is exciting in that we will have a new treatment for Delta, hopefully within the next 12 to 18 months, which had nothing really before. Up until now, we had little data on what is happening with Delta in Canada. And this was an important study that we did through the Canadian Hepatitis B Network. And this map, which was presented at the HBV meeting, showed like blank, no data available for all of our country for hepatitis Delta. So Carla Coffin, she's the senior scientist at the National Microbiology Lab. They do all the testing for hepatitis Delta in the country. She looked at 7,000 samples tested over a 5-year period. Overall, 5% were Delta antibody positive. Keep in mind that though this is a referred population, right? So it's coming from specialists sending in their samples for testing. 65% of them were viremic or RNA positive. A high representation of them were Canadian born, but also a significant immigrant population as expected. And people that were Delta co-infected compared to our mono-infected cohort had much higher risk of end-stage liver disease, cirrhosis, liver cancer, needing liver transplant. So it highlights the importance of us trying to find out about Delta within Canada, because there hopefully will be new treatments coming through the pipeline, and also will reduce the risk of them developing all these very severe complications. Okay, if there are no questions, I'll move on to the second class of drugs. So capsid inhibitors or capsid assembly modifiers. So there are two main classes, one that just block formation of the capsid or encapsulation of the viral RNA. And I like the word that the scientists use for the second term, melting of the capsid, or cause premature black circular DNA uncoating of newly synthesized one. A lot of different clinical trials in phase 1, phase 2, phase 3, overall shown to have a strong antiviral effect, very well tolerated, been used in combination with the approved treatments, the nukes and the interferon, and shown synergy, but no cure, at least with the monotherapy, and did not achieve surface antigen loss. And in some of the early generation of capsids, it's developing resistance. So they need more potent capsid inhibitors or combination treatment. And I'll show you this slide just to highlight how dynamic the field is. Like there was this drug Vebicor, everybody was all excited about. And then like you look at the news wire, right? Flop, flop, you know, kibosh the drug, everything is ending as everything is ending. Another drug that was pretty well advanced as well was found to have drug-induced liver injury and severe ALT elevations. Stop that drug. So they're proceeding, Assembly, the company, is proceeding with a fourth agent that hopefully in 2022 will be doing additional phase 1 or early clinical trials. Moving on, hopefully I'm not going too fast. The nucleic acid polymers, or NAPs. This is an interesting drug and is actually being developed by a Canadian company based out of Montreal, Replicor. You guys might know of this. So NAPs block secretion of surface antigen from cells. And I already explained how the virus has this interesting step where it makes this abundance of surface antigen as immune decoy and contributes to immune cell exhaustion. This was a paper published in Gastroenterology last year, a phase 2 study. It was only done in Eastern Europe in Moldova. And they looked at the combination of the NAPs with tenofovir and interferon, and I'll explain that a little bit more in the next slide. All of the patients were treatment naive with relatively lower levels of viral load, genotypes A and D. There were a number of editorials published about this paper when it came out. I know that it was a little bit controversial from the company's standpoint. There was questions about safety, right? Like if you're preventing all of this surface antigen from leaving the host cell, what is it doing to the hepatocyte? How is it stressing it? The company's argument, or the scientific argument, was that it actually is degraded by the normal sort of lysosomal degradation pathway. There were ALT flares that occurred, but it wasn't clear, at least to the scientific community reviewing it, what was the type of flare, whether it was a host immune-mediated or a viral-induced flare or a drug-induced liver injury flare, because they didn't really do detailed immunological analysis on the T cells and B cells on these patients that received the drug. And so it was unclear exactly about the mode of action with these flares. And there was some questions about what the optimal regimen. But nevertheless, this was exciting data, and it showed like functional cure that was durable, and the company's proceeding with the further development of this agent. So this is an overview of the study design, and please, you can refer to the paper for more details. There were two groups. So the first group was lead-in with tenofovir, then control, so versus triple combination with the NAPs for 24 weeks. And then the part B was the experimental crossover for 48 weeks with the triple combination, showed very good decline in surface antigen. And at the end of follow-up, a high percentage of them were long-term responders with very low level quantitative surface antigen. So certainly impressive results with this drug. Okay, so moving on to the last sort of directly acting antiviral agents. Hopefully I'm keeping on time. So that siRNAs, or RNA inhibitors, are antisense oligonucleotides. So they cause the Hep B RNA to be degraded, or they block production of viral protein, as you can sort of see in my little cartoon. Hep B has another interesting thing. If you're a virologist, you probably appreciate everything it does, like the beauty of Mother Nature, in that it has overlapping genomes. And if you can create some of these siRNAs that can target multiple viral gene products. So in this situation, including surface, can include integrated virus, as well as the X protein, which is key for maintaining HBV cccDNA. So this has been in a number of clinical trials by a number of different industry, phase 1, and phase 2. One in particular that was presented at EASL, I'll talk about in the next slide, had shown good efficacy in terms of decline of viral markers, as well as durability. And plan was to do future studies, of course, and incorporate more immunoanalysis in this. The durability was a little bit surprising in that the company didn't quite understand why it was so profound. There was some question about whether it was activation of the HPV-specific immune responses or persistence of the siRNA in the cells, although this is not thought to be likely because I think from a virologist perspective, this is supposed to be gone after you deliver the drug. So this is the study presented at EASL. So it's a company, a small biotech company. I don't know, Ver or something. And their drug is 2218, was an X-targeting RNA therapeutic, and they presented their week 48 follow-up results. This is the study design, 2 subcutaneous doses, E antigen negative, E antigen positive. They were observed for 12 weeks and then did the virological and other safety assessments. And what they found was that 2 doses given 4 weeks apart were well tolerated, and there were very substantial reductions in surface antigen in both the E antigen negative and E antigen positive participants, but acknowledging 18 and 6 patients is not exactly a large group. But there was a sustained reduction even a year after the last dose, which was surprising. There was speculation that maybe the drug is silencing the cccDNA as well as integrated virus. And this certainly points towards encouraging results and continued development as part of combination regimens. I believe Gilead has talked to this company about doing a combination trial next year, and I'll talk about that when we mention some of the Gilead drugs. Okay, so I'm going to move on to the second class of drugs, which is immunotherapy, and I'll also take a little break here and see if anybody would like to ask any questions.
Yeah, Carla, there's been a couple of questions along the way. Maybe Daryl Tan, one of our infectious disease folks from Toronto, is wondering about the epidemiology of hepatitis D in Canadians living with hepatitis B.
So that's a great question. And unfortunately, we really don't have a lot of data about the prevalence. As I said, from Carla Oshaway's referred study of 7,000 patients, there were 5% of the total 7,000 diagnosed with Delta antibody positive. But I mean, if you look at our Canadian guidelines, we acknowledge the fact that only 1 lab in the country did Delta testing. So we said, refer those with certain specific characteristics for testing. But if you look at the EASL guidelines, they say everybody who's surface antigen positive should have a one-time Delta antibody test. And that's one of the reasons why we're trying to advocate for the provincial lab level. We've applied for funding to do a study in partnership with the different provincial labs. I think Quebec always seems to be ahead of the curve for most of us. They're going to start doing Delta routine testing or offer it as a service. So maybe we'll have more information about the epidemiology and prevalence.
Great, thanks. I guess for people and for clinicians out there, what are the things that you would look for that would make you say, aha, I wonder if they have Hep D, and then you would go to get the testing done?
Yeah, and that's great. It's very interesting that there are certain regions of the world that Delta is highly prevalent. In Mongolia, they have found studies that 60% of surface antigen positive people have Delta. It's been high prevalence in Eastern Europe, in sub-Saharan Africa, in South America. Historically, Delta wasn't thought to be present in Asia, like in Chinese or Taiwan or Japanese, but in my practice and other people have found that it is present within Asia. So I can't say that's 100% correct. If you have somebody who's young with advanced fibrosis, but they have low-level HBV DNA, and you don't quite understand why, that's a good person to check for hepatitis Delta. So, because of viral interactions, Delta can suppress the hepatitis B, and that might give you a clue that it's not related to their Hep B. So let's check for Delta. If you have availability of quantitative surface antigen testing, you might see this profile where there's high levels of surface antigen, low levels of HBV DNA, and then you might think of Delta because Delta uses surface antigen to complete its life cycle.
Great, thanks, Carla. And I guess while we're on the topic, Troy Grennan has asked, would you recommend screening for Hep D if someone who cleared surface antigen, e.g. after many years of chronic hepatitis?
Yeah, in theory, that is very much possible, right? Because they cleared surface antigen, but they might still have like integrated virus or, but I don't think that would be done as standard of care. I would only do it as part of a clinical research project because there's nothing out there documented. But I've seen some like case reports and I've seen some lab stuff, like some preclinical and research stuff about it.
Okay, great. Maybe in the interest of time, we will keep on going with the rest of your
Excellent.
slides.
Let's move. Okay. So immunotherapy, I don't have as many slides because they haven't been as well developed. So as we mentioned, all of the directly acting antiviral agents in terms of entry, interference, core inhibitors, they only target an intermediate step. And if you stop the drug, the virus will relapse. So, at the HPV meeting, there's almost two camps. They talk about you need a viral cure or you need an immune cure. So the immune people say the immune system is the most important to target. It targets the cccDNA because if you kill off the infected liver cell, you get rid of it. It can target integrated virus and viral replication. The problem is that if you're inducing an immune response, then you don't want to have like killing for a cure, right? You don't want a very severe ALT flare and induce, decompensation. Definitely immune-based therapy, you have to be very careful in somebody that has cirrhosis or advanced liver disease. And you also need to differentiate between drug-induced liver injury. You guys know this very well, I'm sure, from your HIV patients, and that you can get ALT elevations related to that. Okay. So summary, the mantra is that a well orchestrated and strong immune response has really driven attempts for these immune mediated immunomodulatory drugs. So we know that the innate immune response is key for sort of recognition of the pathogens, and that the adaptive immune response in terms of coordinated and effective T cells and B cell response is important for clearing infected cells. So these different drugs that have been developed include those that induce innate immunity or toll-like receptors that have been in phase 1 and phase 2, therapeutic vaccines, about 18 published studies or drugs, all in different phases of development. Some of these companies I didn't even recognize. One was from Cuba, as well as more common or certainly larger pharmaceutical companies are looking at. Checkpoint inhibitors, which have been used for cancer chemotherapy, are in early phase development, and others including T cell receptor, NOD2 or RIG-I, that stimulate interferon-stimulated genes. This company, Springbank, actually had progressed quite far along with their drug, and then last year they halted all development because it was found to induce drug-induced liver injury. So again, sort of highlighting how dynamic the Hep B cure field is going. So this is my excuse to show a woodchuck on a slide, on the slide. So the woodchuck hepatitis virus is a very excellent model because you have a functioning immune system. Although the mouse models are very valuable, they can be artificial in terms of seeing the host immune response against a viral infection. So the TLR agonists were developed using the woodchuck model. TLR7 agonist was very far along and showed excellent response in woodchucks, and then when we put it in patients, didn't really work. So Gilead then moved on to their TLR8 agonist, which has shown antiviral effect, was published last year in Hepatology, and they published their phase 2 study at Gilead in EASL, which I'll show next. And they're planning to do a combination of this drug, selvanlutamide, with the siRNA, the one that I just mentioned, developed by this biotech company, Vir, to do that for future cure research, as well as a PD-1 antagonist. So this was a poster at EASL, and in the study they looked at 37 e antigen positive patients, majority Asian because that's our patient population, majority male. They were given 3 milligrams of the drug for 24 weeks along with a nucleoside analog followed by the nuke. Primary endpoint was safety and proportion that had a decline in surface antigen, generally safe and well tolerated, but none achieved their primary endpoint, which was a greater than 1 log decline in surface antigen. A small number had sustained surface antigen decline of about 0.5 log. So this is the reason why Gilead is moving on with this drug, probably in combination, like I mentioned, with the siRNA inhibitor. So it shows promise. And then finally, in terms of the therapeutic vaccines, there are many, and I haven't seen a lot that I think would really be able to highlight right now. And as I mentioned, there are some that have concern about uncontrolled immune activation and fatal hepatitis. So we have to be careful with these therapeutic vaccines. In general, when people were talking about the older vaccine approaches that were used that didn't work in the past, it was because we really didn't have enough suppression of the viral antigen that was contributing to T-cell exhaustion. So if we're doing this in combination treatment with the siRNAs and the nukes that are suppressing viral antigen, then maybe these vaccines will work. So in summary, I just want to highlight many new drugs being developed. There is great hope for a cure. You can tell your patients that. The first goal is functional cure to achieve surface antigen loss. There are many drugs that will likely be needed to work in different ways. And our patients are quite complex, right? Older, younger, E antigen negative, E antigen positive, different immune responses. So we have to figure out what's the right combination for the right patient. I think in terms of the combination, nukes will be the backbone because they're safe, they're potent, they work. Then a capsid inhibitor, one of the scientists said, I will eat my shoe if there's no capsid inhibitor included in this regimen. The siRNAs and antisense oligonucleotides will likely be used. The viral entry inhibitor, which has already progressed and approved for Delta, I think it's going to be a good contribution to our armamentarium. And the NAPs that block surface antigen secretion, although acknowledging there is some controversial editorial comments about it. And then the holy grail is to get rid of the cccDNA. And I like this mnemonic that one of the scientists used: control, combination, and cure. In terms of duration, people think that that you'll need 1 year of treatment in order to get rid of, with this combination, that's the duration that will be needed. But it's important that the drugs work together, they're synergistic, effective, they're safe, and they're affordable. So I don't have a crystal ball, and hopefully this slide won't come back to haunt me when you talk about what's going to happen. So the backbone will be the nukes, and some of the industry, they're looking at continuing with interferon because it's approved and we have experience with it. The next year or so, we hopefully will have Hepcludex-B approved, at least for Delta, and they'll be proceeding with monoinfection. And then in the next 3 to 5 years, all of these new drugs are going to be, some of them, I think at least one of them will be approved. Unfortunately, in context of HIV co-infection, as you guys know, it tends to be the next phase after they approve it for monoinfection. I'm not sure, and maybe the people that were presenting about the immunotherapy for HIV can give me some insight in that. Would you be more likely to want to focus on a DAA versus a therapeutic vaccine? But I know that there's a lot of therapeutic vaccine research being done for HIV. It's going to take much longer time before we achieve the ultimate sterilizing cure of the cccDNA inhibitor. So I'd like to acknowledge all of my colleagues with the Canadian Hepatitis B Network who support all of our efforts. I'd like to acknowledge my coordinators at the University of Calgary, my former coordinator and current ones, as well as our research manager,